Thank you for coming to hear my presentation. This is over my thesis project, and so I observed the genetic changes that are exhibited in Helicobacter or corn earworm caterpillars when they actually feed on several different tissue types of maize, which is our agricultural corn plants. Now, I do my work through the Plant-Animal Interactions Laboratory, and we specifically look to highlight the complex interactions between these two co-evolving organisms, the plants, of course, and our agricultural pests. There's always a constant struggle with any agricultural plant to defend against these herbivores. Molecular work specifically is utilized in our laboratory to highlight these complex interactions, simply because the behavioral and morphological changes exhibited by these two organisms are often so minute that we can't really quantify them as far as analyses. Previous research has determined that plant defenses, because they can upregulate the jasmonate and salicylic pathway against several different types of attackers, the genetic expression or genetic changes are not homogeneous across all different attacker types. They are actually highly specialized, not only to attacker type, but also within different plant species, and also actually within tissues of the same plant. So it's a lot like the human body, where if our left arm is hurt, our right leg is not going to start upregulating necessarily all of those same genes. Now, I do my work with Helicobacter. It's in the Noctuidae family, so don't expect a nice, cute little butterfly. It is actually a moth species, and here it is in the adult form. We do our work because they're easier to contain on the Helicobacter caterpillars, which actually have the chewing mouth parts. It is an agricultural pest found locally around here. It is a generalist, so even though it is named as the corn earworm, where it's usually found on the corn cob, it can actually also feed upon tomato, soybean, tobacco plants. And so it is actually a widely used generalist insect pest in research. The objective of my experiment was to actually feed this Helicobacter caterpillars several different tissues, and I chose our native Illinois maize corn crop, since we are corn country. So I allowed the HZAI caterpillars to feed on several different tissues from the corn plants to observe growth differences, which can be easily determined by weighing them, and genetic expression by using molecular analysis to highlight specific genes that we know of in 3 distinct categories: digestive, growth, and immunity genes, to see if there are certain tissues that stimulate these as compared to just the leaf. Now, because we know that leaves have a quick induction of defensive and responsive pathways, we can assume that the caterpillars, because they're actually normally found in the corn cob or the corn fruit, that the corn fruit and silk tissues where they're usually found should show a smaller impact on growth and a smaller impact on genetic change. So the first thing that I did was actually use a biological assay to determine how well these caterpillars can grow on different corn tissues. We used 3rd instar HZAI caterpillars, so they're still quite in the active feeding stage. And they are placed in isolated solo cups with constant food replenishment. So when we get to the molecular assay, genetic expression is not caused by starvation. It is actually caused by responding to what kind of tissue they are feeding on. So we used a nutrient diet control. This allows the caterpillars to obtain all the nutrients is needed to grow and undergo metamorphosis. And then I used the 4 distinct corn tissues that is found in plants: the corn leaves, the corn husk, the corn silk, and the corn fruit or the corn cob. We initially weighed the caterpillar weight and randomized which caterpillars got into which treatment, and so there was no selectivity. We allowed them to feed for a 72-hour period, again with food replenishment. That's very critical. And then we weighed them a final time after the 72-hour period, and flash froze them in liquid nitrogen to prevent any nucleic acid degradation. The molecular analysis that I used consisted of a real-time qualitative PCR analysis. We used representative genes across 3 different gene types. The first thing we did was extract total RNA using a trizole method and reverse transcribed our mRNA transcripts into cDNA to use in PCR. A nanodrop from Thermo Scientific was actually quite critical in this step because we wanted to normalize our concentrations before we got to PCR. So once again, our gene expression, upregulation or downregulation values was not caused by higher concentrations or lower concentrations. So for my results, from the bioassay specifically, the mean weight in grams, as you can see here on the left, this is my nutrient diet control. Caterpillars who fed on this type of media were allowed to attain more nutrient and grow very rapidly within a 72-hour period. Corn leaf and corn husk here on the right, however, had a very significant detriment to corn growth. But if you can see here, back on the right, corn fruit where they're normally found, while there is a slight detriment because plants can respond even minutely through the corn fruit itself, actually allowed them to grow quite rapidly as well. To go to the molecular analysis, I used actually 14 different genes that I plated. Alpha-tubulin and RPL13 were housekeeping genes I utilized in several plates. These are housekeeping genes that are always on, and so genetic expression, these are our molecular controls. The genetic expression of these two genes do not change unless the organism is near death. And so we can use these genes to quantify relative fold differences in these other genes that we are looking at. Now, the first set of genes I looked at were digestive genes, and so we have trypsin and alpha aminopeptidase, which are proteases that break down plant proteins. It is highly expressed here in the leaf tissue. Not only is the leaf tissue not as nutritive as the corn fruit or corn silk or husk, but also in the leaf because leaves are sites of very high induction of plant defenses. They also have protease inhibitors. So we're having to work twice as hard to get anything out of the plant. Here are three other digestive genes. Alpha amylase digests starch and sugars. Lipase digests lipids. And GADPH actually metabolizes aldehydes. As you can see again, because leaves are not as nutritive and because they are also combating plant defenses and responses in this area, they are having to overexpress genes in order to grow at all. However, we did have a very unexpected result in the GADPH, which metabolizes the aldehydes. Silk, we actually found is very toxic. And again, they are having to overexpress this dehydrogenase to get any nutrient or grow out of this. Now these are growth genes here. We have juvenile hormone-induced protein, JH. We have ecdysone oxidase and prothoracicotropic hormone, PTTH. Juvenile hormone is highly expressed in all 4 plant tissues. Now, originally seeing this result is a little surprising, simply because the juvenile hormone is selectively upregulated only in the caterpillar stages. So this keeps the caterpillars as caterpillars longer. Now this is not really great for the individual plant. Why would you want the caterpillars consistently feeding on your tissues? However, if we think of it as far as an ecological community structure, if we can slow down the reproductive cycles of these caterpillars, and around here we can get about 3 to 5 reproductive cycles in a growing season. If we can consistently slow down caterpillar growth, we might only get 2 or 3 reproductive cycles, meaning not as many caterpillars. So it might not be good for the individual plant, but as far as ecological community, this might be a good community response as far as plants. Now, ecdysone oxidase actually controls molting stages. Now, the caterpillars have to upregulate, again, this molting transition hormone in the leaf simply because they are not growing as fast. And so if they want to become adults, they actually have to overexpress this gene. However, the control in the fruit are upregulated a little bit, but not as much as the leaf here. Now the pro-thoracotrophic hormone, PTTH, is actually the neurohormone, so it's a backup gene that we looked at, that is responsible for upregulating both JH and ecdysone oxidase. Now we did look at 3 salivary glands. These are salivary genes. These genes are secreted in the saliva. The well-known one we used was glucose oxidase. This suppresses plants' defenses as well as glucose dehydrogenase. Now glucose oxidase is highly expressed in the leaf tissue simply because that is the site where responses are very quickly induced. And so they, especially caterpillars, feeding on this tissue want to suppress that as long as they can. Now glucose dehydrogenase is actually also a plant suppressant, but it also breaks down sugar, so it is a digestive gene as well. So we were not surprised to see it upregulated in fruit. Now lysozyme is actually an antimicrobial gene against pathogens such as bacteria and fungi. And now fruit is the site of where there's lots of starch and free sugars. As they're chewing through this material, they're exposing this to the air where pathogenic fungi and bacteria can actually take root. And so to prevent infection of their own body, they will have to overexpress this gene in that area. And the last gene I looked at was Cytochrome P450. This again is a detoxification gene. It is highly upregulated in the silk, and that's because silk is very toxic to caterpillars. To conclude my work, I'm actually inviting more research into looking at different tissues. Because there is a reason Mother knows best in nature, these caterpillars are found within tomato fruits, corn fruits for a reason. That's because they're more nutritive and there's less responses by the host plants in these tissues. And so agriculturally, when we go to use pesticides or increase genetic modification of tissues, we're probably looking at the wrong area. And so with the reduction of the use of pesticides and our outcry against GMO crops, we're going to have to come up with some kind of novel ideas and maybe directing responses against these plant herbivores in these tissues might be an alternative. And I'd like to thank my advisor, Dr. Richard Musser, and the Plant Animal Interactions Lab at Western. And I'd like to invite any questions.